Hi, I'm Natalie Robertson, and thank you for tuning in to Suffolk's McNair and Ballotti Undergraduate Research Symposium. I am a senior in the biology department. I am also a McNair scholar and an ambassador for the program. I had conducted this research last summer, 2020, under the supervision of Phil Stranwitz at Holobiome, where I'd just gotten an internship. But as you may have guessed, this is a computational project because we were online. So without further ado, I'll teach you about some of the computational biology that I learned last summer. And this is my project on the in silico assessment of antimicrobial resistance in consumer probiotics. You may know, bacteria can harbor antimicrobial resistance on plasmids, genomic islands, and mobile elements, and this can act as a reservoir for pathogens because bacteria have the ability to do horizontal gene transfer. So regulatory bodies try to combat this, but often members of genera that are historically considered safe for human consumption, such as Lactobacillus or Bifidobacterium species, may be grandfathered in for having a history of safety. Regulatory bodies also require minimum inhibitory concentration testing in vitro for antibiotics, all of them which would be clinically relevant for the EFSA, the European Food Safety Authority. It is ampicillin, vancomycin, gentamicin, kanamycin, streptomycin, erythromycin, clindamycin, tetracycline, and chloramphenicol. So they require minimum inhibitory concentration testing. But the FDA allows for a self-notification of safety process for probiotics or other supplements on the market. And this means that companies don't have to disclose safety data, and they don't have to get an FDA opinion to be sold in the US. So that led me to ask the question, because safety analyses are conducted by for-profit panels, companies can self-notify safety and bypass the FDA opinion, and many strains are available on assumptions of historical safe use, pre-genomics. So is it possible for acquired antimicrobial resistance genes to exist in commercially available strains? I started by asking this question and went to the internet and used some in silico tools, which were all public access, free, and I think maybe for a few of them I had to make an account, but NCBI, National Center for Biotechnology Information, is a wonderful resource. I acquired 29 genomes from consumer probiotics, or probiotics that are on the market, and I actually sent 12 emails asking for another few strains from manufacturers, but unfortunately I was denied. Those .fasta files from NCBI, the 29 of them that I gathered, I worked with, and analyzed them through the Patrick Comprehensive Genome Analysis tool, which uses a basic local alignment search tool to separate specialty genes, functional categories, quality of the genome, and tell you about the strain phylogeny. So what is this bacteria related to? And Patrick uses the CARD Comprehensive Antibiotic Resistance Database for their antimicrobial resistance outputs, and this is the best. It is a manually curated database of reference DNA, protein sequences, and mutations that contribute to the molecular basis of AMR. And CARD can also let us know if this antimicrobial resistance gene is intrinsic, mutation-driven, or acquired. And acquired is the one that we really don't want. So this image shows you all the genera that I'm looking at and the species within each. A lot of these may look very common because you probably see them on your yogurt or other probiotics that you buy. And then I used PROCA, the Rapid Prokaryotic Genome Annotation tool, to turn those .fasta reads into contigs so they can be sorted. And I got GenBank type files out of PROCA that I put into Island Viewer 4, and this is a tool used to predict genomic islands, which are regions that disproportionately equip microbes with antimicrobial resistance and virulence genes These regions are likely acquired from horizontal gene transfer, and the algorithm studies this by identifying regions where the percent of GC content in a microbe varies from what we would expect it to. Every microbe has its own GC content, and when there's a region that differs from the rest of the genome, this may be a remnant from a horizontal gene transfer event. So from these outputs from Patrick, using the CARD antibiotic resistance database, I found one instance of active resistance. The rest of these in the table under gene have to be mutated to confer resistance to the microbe, so they're not a concern for transmissibility. But on the very bottom, in the Enterococcus faecium T110, there was an aminoglycoside resistance gene, an aminoglycoside acetyltransferase that was found, and the rest of them, I have the L. reuteri strain DSM that the safety dossier addresses the TET-W gene on the plasmid and claims that it was removed by genetic modification, which is possible. It's possible that this gene was only partially removed and the prediction tool still flagged it as TET-W. So perhaps in vitro data would be needed to find out what the case is on that one. The rest of the TET-W genes and all the other strains were addressed in their safety dossiers, and their data shows that they aren't transferable between microbes, so they're also not a safety concern. But that Enterococcus T110 has an aminoglycoside modifying enzyme that's of concern because gentamicin, kanamycin, and streptomycin are all clinically relevant antibiotics that fall under the category of aminoglycosides. So that one, we should collect in vitro data to determine if it's transmissible. And this product is actually not available in the United States, but it has been in production in Japan since 1941. It's very possible that predates genomics, and it's possible there is an antimicrobial resistance gene there, but we won't know until we do an in vitro study. In conclusion, using in silico methods to do gene predictions is very helpful, but it is limited by our knowledge of the molecular basis of AMR. It is limited by the quality of the prediction tools that we use and can be used as a prediction method to direct our in vitro minimum inhibitory concentration analyses. Therefore, it is still a value to have in a system. It is a cost-effective way to assess safety as the probiotics market grows. The probiotics market is expected to grow by 50% just in the next 5 years. So there will be a lot of new probiotics that are making it their way to the market. And this allows us to interpret larger quantities of data when we use computers. We can automate things through in silico methods. It can be very cost and time effective. And when we contribute to these prediction methods, we are improving the quality of analyses by contributing to the data curation of the databases that these tools feed from. In addition, whole genome sequencing contributes to surveillance of antimicrobial resistance, which is a real issue in this time. And increasing publicly available information increases the assuredness of safety in consumer products. So, thank you to Dr. Jarvis Chen and the Suffolk McNair program and the staff and peers there for supporting our research and encouraging us as growing scholars. And thank you to everyone at HoloBiome and Phil for overseeing me as my mentor on this project. Thank you for listening. And if you have any questions or want to read the full paper, please email me. It is nrobertson@su.suffolk.edu. You can add me on LinkedIn. And if the comments in this video are turned on, you can also comment there. So thank you for listening.